Under an endless sheet of ice, the sun barely pierced the frozen world, casting a dim glow over oceans trapped in crystal. This was the Cryogenian, a planet locked in silence, where temperatures plummeted and life clung to existence in hidden refuges. Microscopic organisms thrived in brine veins, geothermal vents, and mineral-rich cracks, crafting networks of survival invisible to the naked eye. Oxygen dwindled, light vanished, and yet life endured, adapting in ways that would shape the future of Earth's biosphere. Step carefully into this frozen expanse and witness the resilience of life against the ultimate chill. Beneath the white silence, life in a frozen world. Across the continents and oceans alike, a white shell encased the planet, sealing every horizon beneath frozen armor. The sun's light, once shimmering across waves and clouds, now reflected from ice, returning most warmth to space. For tens of millions of years, the planet remained in this spectral quiet, a world of reflection rather than absorption, of silence rather than song. The atmosphere thinned with cold, the oceans locked beneath an unbroken crust. Even within that frozen dominion, life endured small, patient, and hidden from view. Beneath the surface, within thin veins of brine and fractures of ice, colonies of microorganisms found sanctuary. Cryogenian formations in Namibia and Australia reveal isotopic anomalies hinting at carbon cycling persisting despite global glaciation. Oxygen isotope ratios in trapped bubbles suggest localized metabolic activity, faint traces of life in an otherwise frozen chemical landscape. The surface above was a frozen desert, its winds sweeping fine crystals of ice across kilometers of featureless plain. Yet under this frozen desert stretched an oceanic abyss, sealed but restless beneath the weight of ice. At depths where geothermal energy still seeped through the crust, the ocean remained liquid, supercooled, dark, and heavy with dissolved minerals. In these deep layers, tiny prokaryotic cells drifted, feeding on molecules released from the slow decay of the Earth's interior. Carbon isotopic shifts in ancient sediments indicate that methanogenic and sulfate-reducing bacteria were not only present, but thriving in microscopic refuges of warmth, their metabolisms continuing even at temperatures near freezing. The ice above was neither solid nor uniform. Microscopic fissures, air bubbles, and brine inclusions ran like threads through the frozen crust, forming what scientists call cryoconite networks, microhabitats, where water, salts, and trace nutrients lingered in perpetual twilight. Modern analogues on Antarctica's Taylor Glacier reveal living microbial consortia in brine channels as narrow as a human hair, metabolizing slowly but continuously. Some draw energy from light filtering through thin ice. Others live off nitrate or sulfur compounds. Their DNA fragments, preserved for millennia, mirror what may have once existed in the Cryogenian seas. Communities bound by chemistry rather than abundance, surviving not by growth but endurance. Beneath what is now the Sahara Desert, outcrops of ancient cap carbonates record the boundary of the Great Freeze. Within them lie stromatolitic textures, layered mineral structures once produced by microbial mats. Their presence beneath and between glacial deposits reveals a paradox. Life had persisted during the freeze itself, entombed within subglacial melt zones or thinly ice-covered waters. These microbial fabrics, found across Namibia, China, and Canada, bear the signatures of photosynthesis, even at a time when sunlight was nearly extinguished. Chlorophyll-derived biomarkers, known as porphyrins, detected in cryogenian rocks, prove that photosynthetic organisms clung to the few translucent refuges where light could still penetrate the ice's thickness. The equator may have offered narrow seas where direct sunlight thinned ice to a few meters. Computer models of planetary albedo suggest that such equatorial belts might have formed dynamic slush ball regions, patches of semi-frozen water sustaining surface photosynthesis. Cyanobacteria, ancient architects of oxygen, likely persisted there, maintaining faint atmospheric oxygenation. Measurements of sulfur isotopes in cryogenian pyrites show subtle, mass-independent fractionation, an indicator that oxygen, though reduced, never disappeared completely. In that whisper of oxygen, the biosphere held its breath and waited. In the crushing cold far below, warmth still seeped upward from the mantle. Where oceanic crust spread apart, hydrothermal vents spewed clouds of dissolved metals and sulfur. These were islands of possibility amid the abyss. Studies of modern hydrothermal microbes, capable of thriving at 120 degrees Celsius near volcanic ridges, give clues to how life might have persisted during global freezing. DNA of methanogens and sulfur-oxidizing archaea traces back to this era, suggesting cryogenian vent fields as cradles of continuity. Iron isotopic records from formations like the Rapitan Group in Canada support this. Evidence that chemosynthetic metabolisms operated in a world without sunlight. In some places, where pressure fractured the ice, faint light filtered through, scattering across suspended particles in a haunting glow. Within these spectral pools, microbial films may have grown along the undersides of the ice, slowly accumulating organic material. Laboratory experiments in polar analogues show that cyanobacteria can remain active at 17 degrees Celsius when embedded in ice, provided minimal light and thin films of liquid water. Their pigments, phycocyanin and chlorophyll A, allow absorption of the faintest wavelengths of blue light, the only color able to pierce thick ice. In that frozen luminescence, ancient photosynthesizers continued their slow labor, turning photons into sugar one molecule at a time. The Snowball Earth was not uniform. It was a world of contrasts, shaped by volcanic eruptions, geothermal upwellings, and orbital cycles that modulated the severity of cold. Each volcanic exhalation injected carbon dioxide into the atmosphere, but the reflective surface above sent most of that heat back into space. Yet within ice-covered basins, that same gas may have formed microclimates, slightly warmer pockets beneath domed ice, rich in nutrients dissolved from hydrothermal minerals. In these rare sanctuaries, complex microbial communities may have flourished. Evidence of organic carbon enrichment in black shales, dated to the Marinoan glaciation, hints that localized productivity surged even during the most severe intervals of ice coverage. In the geological record, the silence of this age is nearly total. No bones, no shells, no coral reefs, only subtle traces of biochemistry encoded in carbon isotopes and biomarker molecules. Yet within those faint signals lies a narrative of resilience. Life did not need abundance to persist. It required only space enough for chemistry to continue, time enough for one cell to divide into two. Beneath the kilometers of ice, the biosphere transformed from an empire of abundance into one of endurance, refining the art of survival at the molecular scale. Enzymes evolved to function at freezing temperatures, membranes grew thicker to resist rupture, and genetic systems learned to repair the slow damage of radiation that filtered through the translucent crust. Every molecule of carbon that passed through these cells was an act of defiance, a continuation of life against thermodynamic odds. Each metabolic reaction was a whisper in the darkness, a reminder that Earth's vitality could not be extinguished by cold alone. Over time, sediments accumulated at the base of glaciers, incorporating dead cells and metabolic residues into mineral layers. These became the archive of that frozen age, preserved in stone for future scientists to read. In their isotopic fluctuations and chemical signatures, we hear the heartbeat of that silent world, a rhythm so slow, yet so unbroken, it outlasted the age of ice itself. And when the world above remained frozen and still, the depths below continued their faint respiration, powered by warmth from the core and light diffused through miles of ice. The surface told a story of lifelessness, but the earth beneath whispered otherwise. Beneath that vast white silence, in brine-filled pores and volcanic shadows, the continuity of existence held firm. There, unseen and uncelebrated, life outweighted the planet's coldest test. For in the deep places where the ice met stone, the heat of the earth still lingered. It was a quiet warmth, steady and unseen, carrying with it the first promise that the planet's silence would not last forever. Beneath that frozen stillness, life waited patient, unseen, and ready to rise again when the world remembered the sun. The fire beneath the ice, oases of survival. Below the unyielding ice, the planet itself beat with secret life. Where glaciers pressed against the crust, fractures and subglacial lakes allowed molten heat to seep upward, creating pockets of liquid water in an otherwise frozen abyss. From these hidden fissures, volcanic vents expelled mineral-rich fluids, forming chemical islands in a world of ice. The heat and dissolved compounds created oases where life could thrive, even in the absence of sunlight, providing both energy and nutrients to microbial communities that had survived the cold by waiting. Modern analogues from mid-ocean ridge hydrothermal systems show dense colonies of thermophilic archaea and bacteria clinging to vent chimneys, converting inorganic chemicals into the building blocks of life, a process mirrored in the Cryogenian oceans millions of years ago. The chemistry of these deep refuges was extraordinary. Sulfur, iron, hydrogen, and methane coursed through vent fluids, sustaining metabolisms independent of photosynthesis. Isotopic analyses of Cryogenian black shales reveal signatures consistent with chemosynthetic activity, suggesting that life exploited every available gradient of energy. Methanogens converted carbon dioxide into methane. Sulfur reducers transformed sulfates into hydrogen sulfide. And iron oxidizing bacteria precipitated minerals that would become fossilized in future sediments. These processes did not merely support survival, they created thriving micro-ecosystems in which chemical energy flowed like a hidden river, providing both sustenance and the potential for evolutionary experimentation. Life adapted not only chemically, but physically to its volatile home. Thermophilic microbes, similar to modern vent-dwelling species, evolved enzymes that remained stable at temperatures approaching 120 degrees Celsius. Membranes developed increased rigidity to withstand chemical and thermal stress, and molecular chaperones preserved protein structure under extreme conditions. Fossilized iron sulfide deposits, some exceeding meters in thickness, indicate dense microbial mats once clung to these vents, leaving traces of collective activity etched into the rock record. Each mineral layer represents countless cycles of metabolism, a testament to life's persistence against overwhelming odds. Volcanic eruptions and hydrothermal pulses punctuated this otherwise dark world. Pyroclastic flows beneath ice may have melted kilometers of frozen water, temporarily expanding habitable space for microorganisms. Geochemical gradients shifted rapidly, selecting for flexible metabolisms capable of switching between electron donors and acceptors. Studies of modern Antarctic subglacial lakes demonstrate that microbial populations can respond to sudden nutrient influxes with rapid bursts of activity, a pattern likely mirrored in Cryogenian refuges. These oases were not static, they were dynamic laboratories of survival, shaping evolutionary potential within isolated, extreme habitats. Some microbial colonies formed towering mats along vent structures, layering themselves like miniature cities in miniature darkness. These biofilms captured dissolved minerals, influencing the local chemistry and providing structure for other cells to attach. Their growth was slow but relentless, sometimes leaving impressions in precipitated sulfide and carbonate deposits that survive to this day. Examination of the Rapitan group in Canada and similar formations in Namibia reveals that such layered structures correlate with isotopic evidence of microbial metabolism, indicating that these tiny architects maintained a rhythm of life even as the surrounding planet froze solid. The distribution of these oases was uneven. Not every fracture or vent provided sufficient warmth or chemical energy. Some regions of the Cryogenian ocean remained completely lifeless, sealed beneath kilometers of ice. But wherever geothermal activity intersected with liquid pockets, life flourished, producing local biodiversity unmatched elsewhere on the frozen planet. The contrasts between barren expanses and productive vent fields underscore the resilience of life and the creative potential of extreme environments. Even without light, these oases sustained carbon cycling, sulfur transformations, and metabolic interactions that kept the biosphere coherent and persistent. Adaptation in these refuges extended beyond simple survival. Horizontal gene transfer among microbial populations accelerated innovation, allowing beneficial traits such as cold-stable enzymes or enhanced electron transport chains to propagate quickly. Genetic evidence from modern extremophiles suggests that many of these adaptations trace their origins to Cryogenian ancestors, indicating that the evolutionary pressures imposed by ice and chemical scarcity produced a durable lineage of resilient life. Each microscopic innovation strengthened the biosphere, preparing it for future challenges once the ice began to thin and light returned. Despite the isolation and harshness, these oases were hubs of chemical communication. Gradients of hydrogen, methane, and sulfide shaped the interactions between species, creating microecosystems that were self-organizing and interdependent. Microbial mats facilitated nutrient recycling, producing compounds that diffused outward to support other cells clinging to less hospitable niches. The chemical networks in these hidden refuges, though invisible to the human eye, were complex and highly efficient, sustaining life in a world that might otherwise have seemed utterly inhospitable. The geological record confirms the presence of these vibrant, heat-driven ecosystems. Ancient hydrothermal deposits, marked by distinctive sulfur and iron isotopic ratios, indicate that life flourished wherever energy and liquid water intersected. Trace fossils, mineral precipitates, and isotopic anomalies provide windows into an ecosystem that operated in darkness and heat, isolated from the frozen oceans above. Each data point illuminates not only survival but also innovation, the slow crafting of biochemical systems capable of sustaining life for millions of years under extreme conditions. Even in the coldest reaches, the Earth's interior refused to yield completely to the ice. Hydrothermal oases provided warmth, chemical richness, and opportunity for life to expand. Microbial communities metabolized sulfur, iron, and methane with patience and precision, leaving signatures that would endure in rock for eons. They became crucibles of evolution, holding the spark of life that could someday reach the surface again. In this hidden warmth, Earth's biosphere pulsed quietly, a reminder that vitality persists wherever energy flows. In the shadow of endless ice, life endured, embracing both protection and potential. Each vent and subglacial lake was an island of activity in an otherwise immobilized world, a place where chemistry and heat intertwined to sustain communities. These oases illuminated the remarkable capacity of life to endure in the face of planetary-scale extremes. From the glowing heart of the Earth, energy and survival intertwined, hinting at the next chapter, where even the faintest sunlight would reach through the icy equator and allow life to reach toward light once again. The dim belt of life, sunlight beneath the ice. Beyond the glowing vents, a faint light filtered through thinner stretches of equatorial ice, offering a fragile promise of survival. While most of the planet remained encased in kilometers of frozen crust, these narrow belts allowed the first photons to penetrate, reaching the water beneath, with enough intensity to sustain the most resilient photosynthetic life. The equatorial regions, warmed ever so slightly by direct sunlight and thinner ice, formed a patchwork of potential oases where microbial films could cling to existence. Climate models of cryogenian ice sheets indicate that these areas may have persisted for tens of millions of years, creating a continuous but precarious corridor of light-dependent life amid a global freeze. Microscopic cyanobacteria formed delicate films along the undersides of the frozen surface, harvesting photons that had travelled through kilometers of ice. These organisms, some only a few micrometers in diameter, used pigments specifically adapted to absorb the faint blue wavelengths capable of penetrating the thick ice. Laboratory studies of modern Antarctic analogues demonstrate that cyanobacteria can photosynthesize under ice up to 12 meters thick, albeit at greatly reduced efficiency. Fossil evidence from cryogenian cap carbonates, including preserved acritarchs and organic biomarkers such as porphyrins, suggests that these equatorial microbial communities were widespread and played a pivotal role in sustaining oxygen fluxes even during the height of glaciation. Isotopic evidence in cryogenian carbonates indicates that even these tiny photosynthetic communities contributed small amounts of oxygen to the atmosphere. Variations in delta to the power of 13C and delta to the power of 18O ratios reveal localized production and sequestration of organic carbon, hinting at a subtle but persistent photosynthetic pulse. This limited oxygenation may have influenced biogeochemical cycles in ways that supported other microorganisms in adjacent dark zones, creating microgradients of redox potential that allowed life to exploit every niche. Though modest in scale, these contributions demonstrate that even under extreme global cooling, Earth's biosphere continued to exercise influence over planetary chemistry. Slender channels of brine threaded through the ice, delivering nutrients to these microbial films in the dim equatorial belt. Saline inclusions, enriched with nitrate, phosphate, and trace metals, sustained metabolic processes that would otherwise have been impossible in the frozen waters. In modern polar environments, brine channels support active microbial colonies despite sub-zero temperatures, providing a model for understanding how cryogenian cyanobacteria could maintain minimal growth and reproduce within these isolated pockets. The combination of light penetration and nutrient delivery formed a delicate equilibrium, sustaining life against the oppressive stillness of the Snowball Earth. Climate models suggest that seasonal thinning of the ice created narrow, fleeting windows of light that life could exploit. During periods of slightly higher solar insolation or transient warming from equatorial volcanic activity, ice surfaces may have temporarily thinned to mere meters, allowing higher intensity photons to reach microbial mats. These pulses of light likely triggered short bursts of photosynthetic activity, which, while minor in the global energy budget, were critical for sustaining populations in the long term. Fossil evidence of laminated microbial mats within cap carbonates supports the hypothesis that life was not uniformly dormant, but dynamically responding to environmental fluctuations. Within these fragile ecosystems, organisms competed for scarce energy, developing strategies to optimize light capture in near darkness. Cyanobacteria arranged themselves into films that maximized surface area and exploited even the faintest scattering of light, while others adopted dormant states to survive extended periods of darkness. The interplay of competition and cooperation shaped community structure, creating resilient networks capable of weathering harsh environmental oscillations. This subtle choreography of survival demonstrates that even microscopic life can exercise agency, engineering microhabitats in response to environmental constraints. Organic biomarkers preserved in sediments reveal adaptations such as low light pigments and enhanced photosynthetic efficiency. Porphyrin derivatives and carotenoids extracted from cryogenian rocks indicate that cyanobacteria and possibly early algae had evolved mechanisms to exploit minimal energy inputs. Such adaptations not only ensured survival, but also facilitated incremental contributions to atmospheric oxygen levels, setting the stage for later biological innovations. The persistence of these chemical signatures across geographically distant sites suggests that light-driven refuges were widespread along equatorial belts, forming a connected network of tenuous productivity. The delicate balance between light and shadow shaped these communities, making every photon a resource for survival. Microbial mats in the equatorial ice acted as living lenses, absorbing sunlight while redistributing nutrients and dissolved gases. Their activity created microscale oxygen gradients, fostering localized ecological interactions that would ripple through the subsurface biosphere. In this interplay of scarcity and efficiency, life demonstrated its remarkable ability to engineer stability within extreme environmental constraints, maintaining coherence across a planet otherwise dominated by ice. Even in these faintly illuminated refuges, microbial metabolisms left chemical footprints that hint at oxygen production beneath the ice. Sulfur and carbon isotope anomalies in cryogenian sediments reflect the delicate balance of photosynthetic activity and respiration, while mineralogical evidence points to ongoing precipitation of carbonate and silica within microbial mats. These signatures confirm that equatorial light belts were not passive bystanders to the freeze, but active participants in the chemical and biological dynamics of the planet, quietly sustaining life at the margin of possibility. As the light faded toward the poles, life relied entirely on chemical energy, preparing for the descent into darkness that awaited in the frozen oceans beyond the equator. The equatorial bands offered hope and continuity, yet beyond them lay vast expanses where photosynthesis was impossible and survival depended solely on chemosynthesis or metabolic endurance. The dim belt of life, suspended between illumination and shadow, reveals the ingenuity of organisms adapting to the extremes of Snowball Earth, bridging the warmth of hydrothermal refuges below and the absolute darkness of ice-covered seas beyond. In these narrow, fragile corridors, life had learned to persist, teaching the planet that survival was not a matter of abundance, but of adaptability. The black vents of creation, where sunlight never reached, the ocean floor simmered with unseen energy from deep within the earth. In the abyssal plains of the cryogenian oceans, darkness was absolute, yet hydrothermal activity provided localized warmth and chemical sustenance. Vents and fissures expelled mineral-rich fluids, forming chimneys and plumes that created microhabitats for entire ecosystems. Though invisible from above, these deep-sea oases pulsed with life, sustained not by light, but by the Earth's internal heat and the chemical reactions it fueled. Modern mid-ocean ridge systems reveal communities thriving under similar conditions, providing a window into how ancient life may have persisted for millions of years beneath kilometers of ice. Hydrothermal plumes rose from the abyssal crust, carrying heat and minerals into the dark waters. Temperatures around vent openings often exceeded 350 degrees Celsius, yet turbulent mixing with surrounding near-freezing seawater created gradients that microbes could exploit. Dissolved sulfides, iron, and methane became the raw materials for chemosynthetic metabolisms, driving the conversion of inorganic molecules into organic matter. These plumes, invisible and continuous, formed the lifeblood of deep-sea communities, demonstrating that energy could flow independently of the sun and sustain complex biological processes in total darkness. Colonies of chemosynthetic microbes clung to vent chimneys, transforming inorganic compounds into life-sustaining energy. Sulfur-oxidizing bacteria and archaea metabolized hydrogen sulfide, while methane oxidizers utilized hydrocarbon-rich fluids, forming the foundation of trophic networks. Microscopic mats blanketed mineral surfaces, capturing chemical energy and producing biomass that would feed higher organisms. Fossilized vent structures from Cryogenian deposits in Canada and Namibia show layered mineral and organic signatures, indicating long-term microbial occupation and active metabolism in these extreme environments. Black sulfide deposits accumulated around these vents, preserving a fossil record of thriving microbial ecosystems. Pyrite-rich layers and associated carbonate precipitates reveal the intensity and longevity of chemical transformations. Geochemical analyses indicate that these deposits not only recorded microbial activity, but also influenced local fluid dynamics, creating stable microenvironments for life to persist. These mineral formations provide enduring evidence that even in a world dominated by ice and darkness, pockets of vitality could flourish for millions of years. Methane and sulfur cycling powered entire food webs in a world devoid of light, feeding not only microbes but also primitive eukaryotes. Single-celled eukaryotes, possibly early protists, likely grazed on microbial mats, while symbiotic relationships developed between chemoautotrophic bacteria and host organisms. Stable isotope ratios in Cryogenian black shales suggest active carbon and sulfur cycling, revealing that energy derived from chemical gradients could support surprisingly diverse ecosystems. These findings challenge the notion that life under Snowball Earth was uniformly sparse, highlighting instead the pockets of abundance deep below the ice. Within vent chimneys, symbioses linked microbes to mineral surfaces, forming scaffolds that shaped the ecosystem itself. Microbial consortia produced extracellular polymeric substances, EPS, that bound sulfide and carbonate minerals, creating mats and filaments resilient to turbulent flows. Fossilized stalks and tubes from Cryogenian deposits reveal microbes constructing durable frameworks that sheltered themselves and hosted other organisms, from archaea to proto-eukaryotes. Modern hydrothermal analogues, such as those at the East Pacific Rise, show that these structures influence fluid chemistry, local currents, and temperature microgradients, suggesting that Cryogenian vents harbored similarly complex, self-organizing ecosystems unseen in most other environments. Pressure and temperature gradients shaped communities, creating microhabitats that fluctuated with every hydrothermal pulse. Vent fluid chemistry, local currents, and mineral precipitation patterns dictated which microbes dominated each niche. Organisms adapted to extreme thermotolerance, rapid metabolic shifts, and chemical stress, forming resilient networks capable of withstanding the harshest fluctuations. In this hidden world, survival depended on precise adaptation to chemical and thermal extremes, illustrating how life could thrive in total darkness through sheer metabolic ingenuity. Organic biomarkers extracted from abyssal sediments indicate long-term metabolic activity, showing life's persistence in total darkness. Lipid molecules preserved in sulfide deposits confirm the presence of sulfur-oxidizing bacteria and methanotrophs, while isotopic fractionation patterns suggest active growth and reproduction over extended periods. Comparisons with modern vent systems demonstrate that similar metabolic strategies have endured for billions of years, making these cryogenian ecosystems some of the most ancient continuous biospheres on the planet. Even in this pitch-black realm, energy flowed, sustaining networks of organisms that depended entirely on chemical reactions. Microbial mats, vent chimneys, and surrounding sediments formed a cohesive system where metabolites and minerals circulated continuously. Each organism contributed to the stability of the ecosystem, recycling energy and matter in a tightly coupled web. The absence of light was irrelevant. Life's persistence relied on the invisible chemistry of the deep earth, proving that survival could be entirely decoupled from solar energy under extreme conditions. The dynamics of vent ecosystems reveal a complex choreography where survival depended on chemical gradients rather than photons. Pulses of venting, shifts in fluid composition, and fluctuating temperatures created temporal niches that life exploited. Organisms evolved strategies to rapidly respond to changes, from dormancy during chemical scarcity to bursts of activity when nutrients surged. These cycles of adaptation and response demonstrate that even in the most isolated environments, life was dynamic, innovative, and resilient, challenging assumptions about the limits of survival in extreme darkness. From these deep, fire-lit environments, life's resilience echoed silently, a reminder that even in total darkness, existence endured. The black vents of creation reveal ecosystems that thrived without sunlight, sustained solely by heat and chemistry, and show that life's ingenuity extended far beyond visible light. These abyssal refuges formed the hidden backbone of the planet's biosphere, preserving vitality through the harshest conditions and preparing Earth for the eventual return of frozen surfaces to more hospitable states. In the dark, life had found its ultimate testament, that even when the world above sleeps, persistence continues unseen. The sleepers within the stone, hidden under frozen oceans, fissures in stone harboured life in near total stillness. The cryogenian world above was a frozen expanse, yet deep in the rock, microbes found sanctuary in pore spaces and fractures, insulated from the relentless cold. These narrow microhabitats allowed life to endure, not through abundance, but through the remarkable ability to slow time itself. Fossil evidence and mineralized veins indicate that microbial colonies were present across vast stretches of the seafloor crust, suggesting that even in geological stillness, life maintained a quiet pulse, imperceptible yet persistent. Across microscopic pores, microbes suspended their activity, entering metabolic states that could persist for millennia. Known as cryptobiosis or near-suspended animation, these survival strategies drastically reduced energy requirements, allowing organisms to withstand the harshest environmental conditions. Contemporary studies of subglacial rock microbes and deep crystalline crust analogues reveal that some species can repair DNA slowly while remaining nearly inert, providing a template for understanding cryogenian dormancy. In these silent chambers, metabolism continued at a barely measurable rate, enough to sustain essential cellular machinery without depleting scarce resources. Geochemical analyses reveal isotopic signatures of ultra-slow carbon cycling, evidence of life enduring in extreme isolation. Measurements of delta to the power of 13C in ancient silicate-hosted microbial mats suggest that even at temperatures near freezing and with limited fluid flow, carbon fixation and respiration persisted. Trace element ratios indicate subtle but continuous chemical exchanges, hinting at metabolic resilience over thousands of years. These microscale cycles demonstrate that life in Stone Age refuges was not static, but maintained a delicate equilibrium between dormancy and minimal activity. DNA repair enzymes allowed these microorganisms to survive radiation and chemical stress despite minimal energy inputs. Proteins capable of excising damaged nucleotides and restoring double-stranded DNA ensured longevity even as chemical gradients waned. Such molecular machinery would have been crucial in a world where liquid water was scarce and exposure to background radiation was constant. Fossilized remnants of these enzyme systems are difficult to detect directly, but modern microbial analogues indicate that such strategies are essential for ultra-slow survival in extreme environments. Mineral-lined cavities provided both protection and a framework for metabolic dormancy. Layers of silicate and carbonate minerals stabilized pore structures, shielding microbes from mechanical stress and temperature fluctuations. Over time, microbial activity influenced mineral deposition, creating self-reinforcing microhabitats that persisted across millennia. In cryogenian sediments, these mineralized pores serve as evidence that life was not only surviving, but subtly reshaping its environment. Even in the absence of light and abundant liquid water, trapped fluid inclusions within rock offered intermittent pulses of nutrients, sustaining tiny pockets of activity. These microscopic reservoirs contained dissolved ions, organic molecules, and gases that could be metabolized when fluid flow or melting events provided access. Laboratory experiments show that microbes can awaken from dormancy in response to brief nutrient availability, perform essential metabolic functions, and then return to a suspended state. This cyclical pattern of slow activity and dormancy illustrates a delicate interplay between survival strategies and environmental fluctuations. Microscopic fissures channeled chemical gradients, letting microbes access energy hidden within the rock. Redox reactions along mineral surfaces provided a subtle, continuous energy supply, sustaining microbial metabolism for centuries. Iron and sulfur-rich minerals acted as electron donors or acceptors, enabling chemolitho-autotrophs to generate ATP without light or abundant nutrients. Cryogenian mineral veins suggest these chemical energy networks maintained life even during global glaciation, with the planet's crust serving as both shelter and energy reservoir for these sleepers. Modern studies of deep crystalline crust and Antarctic subglacial rocks provide analogues for these cryogenian sleepers. Researchers have found microbial cells capable of dividing once every few thousand years, sustaining ecosystems in environments previously thought sterile. Such findings suggest that cryogenian rock-bound microbes could have survived through entire glacial episodes, persisting until conditions elsewhere began to warm or thaw. These analogues also reveal the limits of life, offering insight into the extreme flexibility and patience required for existence in frozen, isolated refuges. The slow pulse of subterranean water movement dictated which microbes could awaken and reproduce, even briefly. Minute flows through fractures delivered essential ions and redistributed metabolites, creating microgradients that structured microbial communities. Organisms tuned to these fluctuations were able to exploit even the tiniest pulses of energy, maintaining a rhythm that spanned centuries rather than hours or days. The result was a silent choreography of survival where life persisted quietly, almost imperceptibly, in a world dominated by ice. Throughout this quiet world, life persisted with a subtle, enduring strength that redefined survival itself. Metabolism slowed to near invisibility, reproduction occurred in fleeting bursts, and ecological interactions occurred at a geological timescale. Yet despite the apparent stillness, these microbes formed the foundation for future recovery, preserving genetic diversity and metabolic potential. Their endurance demonstrates that life is not defined solely by growth or abundance, but by the capacity to survive across unimaginable spans of time. Silent, stone-bound dormancy points towards saline veins that carried the last liquid whispers of a frozen planet. As life persisted in the rock, pockets of brine threaded through the crust, holding remnants of the ancient ocean and sustaining tiny bursts of metabolism. These veins would become conduits for microbial migration and nutrient flow, connecting isolated sleepers to broader ecosystems as the Snowball Earth slowly began to thaw. Within the stillness of stone, life had perfected patience, offering a quiet testament to resilience and hinting at the delicate revival that awaited the frozen planet. Brine arteries, the frozen veins of life. Through the narrowest fractures of ice, pockets of liquid salt water clung stubbornly to existence. While the planet's surface lay locked in a thick global shell of ice, these brine-filled channels threaded invisibly beneath kilometers of frozen ocean. Even a vein no wider than a human hair could harbor life, forming a labyrinthine network where microorganisms found both refuge and the means to persist. Estimates suggest that cryogenian brines, concentrated by freezing and exclusion of salts, could maintain liquid microenvironments at temperatures as low as 20 degrees Celsius, creating isolated corridors capable of supporting microbial communities for centuries. Fossilized inclusions in ice and mineralized veins provide a lasting record of these hidden microcosms, showing that life endured not through abundance, but by occupying the narrow margins of a frozen world. Saline veins created networks invisible to the naked eye, yet vital for microbial survival. In these passages, tiny microbial mats clung to mineral walls, capturing scarce chemical energy while waiting for subtle pulses of fluid flow. Minor shifts in ice pressure, seasonal warming, or geothermal heat sometimes allowed temporary expansion and reconnection of veins, letting microorganisms migrate or exchange metabolites across isolated refuges. This microscopic connectivity, though ephemeral, transformed the frozen crust into a living lattice, a hidden ecosystem functioning at a scale invisible to most observation, yet essential for survival across global glaciation. Minute fractures channeled gradients of chemicals, letting microbes draw energy from the stone itself. Redox reactions along sulfide and iron-rich minerals created opportunities for chemolitho-autotrophic metabolism, sustaining ATP generation even in the absence of light. Laboratory studies of modern deep subsurface microbes suggest that cryogenian organisms could rely on similar reactions for slow, sustained metabolism over millennia. By leveraging these mineral-mediated energy pathways, life persisted in the darkest, coldest corners of the planet, revealing that the crust itself functioned as both shelter and engine for survival. Halophilic bacteria concentrated their cytoplasm, balancing osmotic pressures against the surrounding brine. Specialized proteins and compatible solutes, such as glycerol and trehalose, stabilized cellular components and maintained enzymatic function at sub-zero temperatures. Membrane lipids were adapted to retain fluidity despite high salinity, while enzymes evolved structural flexibility to catalyze slow biochemical reactions. Modern analogues in Antarctic hypersaline lakes and fluid inclusions show that these adaptations are not merely theoretical. They represent proven strategies for life enduring near-freezing hypersaline conditions over centuries or millennia. Nutrients and chemical gradients for metabolism arose from the interplay of mineral impurities and dissolved ions. Iron and sulfur compounds allowed chemolitho-autotrophs to generate energy, while trace phosphorus and nitrogen sources sustained biosynthesis. Microbial mats forming along mineral surfaces not only captured these nutrients, but subtly influenced mineral deposition, creating microstructures that stabilized habitats and enhanced longevity. Fossil evidence from Cryogenian deposits, including isotopic signatures in sulfide and carbonate veins, shows that these processes were occurring at small scales, yet persisted long enough to shape local ecosystems. Fluid inclusions, locked in rock, delivered sporadic nutrients that allowed life to persist in tiny refuges. Even minuscule fluctuations in iron concentration or temperature could trigger bursts of metabolic activity, allowing cells to repair damage, replicate minimally, or exchange genetic material. These microcycles of activity and dormancy were crucial, as prolonged inactivity could still leave life vulnerable to chemical stress or radiation. By responding to transient resource availability, microbes maximize the utility of these narrow veins, preserving the smallest threads of life until conditions allowed survival to continue. Periodic shifts in temperature and ice pressure altered brine connectivity, determining which microbes could persist or move. Slight warming events or ice deformation temporarily expanded veins, enabling microbial migration or limited colonization of adjacent microhabitats. The resulting network of ephemeral corridors allowed populations to maintain genetic diversity and share metabolic functions, even when separated by long periods of isolation. This interplay between dormancy and transient activity created a subtle rhythm of life that unfolded at a geological timescale where the slowest processes dictated survival. Fossilized brine inclusions in Cryogenian ice cores suggest that these microhabitats endured over millennia. Tiny pockets trapped within the ice preserve both chemical and structural evidence of microbial mats, indicating that life persisted even when the surrounding ocean had solidified completely. These frozen snapshots highlight the extraordinary resilience of life and its capacity to exploit niches invisible to conventional observation, suggesting that Snowball Earth was not a uniform desert of death, but a patchwork of hidden refuges. At the molecular scale, enzymes adapted to intense salinity ensured that metabolism could continue in frigid conditions. Proteins maintained flexibility, membranes retained fluidity, and ATP-generating pathways functioned slowly but reliably, ensuring that essential maintenance processes could occur indefinitely. These biochemical adaptations reveal that life was not merely surviving, but maintaining functional integrity, preparing for eventual reconnection with more hospitable environments as the ice began to fracture and thin. These liquid networks linked isolated pockets of life, creating a hidden circulatory system under the ice. Organisms within brine veins interacted in symbiotic relationships, shared metabolites, and even influenced local mineral deposition. The veins acted as arteries for survival, connecting otherwise fragmented populations, preserving biodiversity, and allowing microbial communities to persist until the eventual thaw. Such persistence demonstrates that even extreme isolation could not completely sever the continuity of life. Even as most of the surface froze solid, these veins acted as lifelines for microbial diversity. They maintained slow metabolic rhythms, ensured genetic continuity, and allowed microorganisms to survive in a hostile, frozen world. These hidden arteries illustrate that survival does not always require abundance or rapid growth. Sometimes, endurance in the smallest niches is sufficient to preserve the foundations of life. Encased in frozen channels, life carried forward the chemical and genetic instructions for future forms. Halophiles and bacteria maintained metabolic activity, however slow, sustaining the potential for future evolutionary innovation. These secret veins not only kept life alive, but preserved the blueprint for emerging eukaryotic cells, hinting at the eventual resurgence of complexity once the frozen world began to thaw. Through these microscopic networks, life endured the Snowball Earth, quietly preparing for a planet yet to awaken. The survivors with a nucleus. Amid the frozen brine, tiny eukaryotes preserved the architecture of life within their nuclei. Beneath layers of solid ice and interlaced brine veins, these cells maintained organized compartments that safeguarded DNA, enabling survival under extreme cold and chemical stress. While surrounding bacteria persisted with simpler mechanisms, eukaryotes carried both the structural and informational blueprints for future complexity, quietly holding the spark of innovation that would one day give rise to more elaborate life forms. Fossil and molecular evidence suggests that these organisms endured for millions of years in isolation, illustrating a remarkable balance between fragility and resilience. Organelles maintained metabolic order even when energy sources were scarce and temperatures hovered near freezing. Mitochondria and chloroplast-like structures enabled minimal but vital ATP production, sustaining essential maintenance and repair processes over centuries. Enzymes adapted to sub-zero conditions allowed the controlled flow of chemical reactions, even in the brine's extreme salinity. This internal organization permitted slow but continuous metabolism, a heartbeat barely perceptible against the stillness of the frozen planet. Some eukaryotes carried tiny passengers within, early symbiotic partners that amplified their survival potential. Certain cryogenian eukaryotes harbored nascent endosymbiotic bacteria or proto-organelles, precursors to mitochondria and plastids. These partnerships allowed for more efficient energy extraction from scarce chemical gradients, supporting slow but sustainable metabolism in the cold, saline brine veins. Evidence from isotopic signatures and structural fossil remnants suggests that such internal alliances may have provided selective advantages, enabling eukaryotes to endure prolonged isolation while maintaining functional versatility in extreme environments. DNA packed into chromatin strands allowed these cells to survive radiation and chemical stress that would cripple simpler organisms. Tight coiling and association with protective proteins prevented degradation from ionic fluctuations and cosmic radiation penetrating the thin ice. The genome itself became a vessel for endurance, storing the instructions needed for replication, repair, and the eventual emergence of complexity. These cells demonstrate that the evolution of a nucleus was not merely a structural innovation, but a survival strategy finely tuned to extreme and isolated conditions. Membrane-bound compartments isolate biochemical reactions, optimizing efficiency under extreme constraints. Enclosed vesicles enabled simultaneous metabolic pathways without interference, providing flexibility that prokaryotes lacked in the frozen microhabitats. Some organelles concentrated essential ions and molecules, creating microenvironments that maintained chemical stability despite the surrounding brine's fluctuations. This compartmentalization allowed eukaryotes to exploit energy gradients and sustain life, even when resources were minimal and intermittent. Within the cytoplasm, a suite of molecular shields preserved function when freezing threatened to crystallize life itself. Molecules like antifreeze proteins, trehalose, and polyols stabilized cellular membranes, enzymes, and nucleic acids against sub-zero stress. These cryoprotectants prevented ice-induced damage, allowing essential biochemical reactions to continue at minimal rates even in hypersaline microhabitats. Comparisons with modern extremophile eukaryotes, such as Antarctic algae and snow-inhabiting protists, reveal that these strategies were highly effective for maintaining cellular integrity across millennia of global glaciation. Mitochondria and other energy processing structures enabled minimal ATP production, sustaining the slow heartbeat of survival. Even tiny fluctuations in iron concentration or temperature could trigger bursts of metabolic activity, and organelles ensured that energy was allocated efficiently. Cells could repair damage, replicate slowly, or maintain homeostasis for centuries, highlighting the remarkable biochemical resilience of early eukaryotes in the Cryogenian ice. This internal efficiency set the stage for evolutionary experimentation, laying the groundwork for the emergence of multicellular life once environmental conditions improved. Cytoskeletal networks provided stability and organization, counteracting ice-induced mechanical stress. Microtubules and actin-like filaments supported intracellular transport and maintained structural integrity, preventing damage as ice shifted or brine veins contracted. This internal scaffolding enabled eukaryotes to remain functional in confined spaces, allowing them to exploit microhabitats that simpler cells could not occupy. By maintaining shape and order, the cytoskeleton contributed to both immediate survival and long-term evolutionary potential. Eukaryotes navigated their confined microhabitats, coordinating subtle exchanges with surrounding bacteria. Symbiotic interactions allowed nutrient sharing and chemical signaling, creating microecosystems that maximize survival in brine veins. Some bacteria supplied metabolites directly to host eukaryotes, while organelles within eukaryotic cells could harness these inputs to maintain ATP production or repair mechanisms. This cooperation demonstrates that even in isolation, life found ways to weave complexity into survival strategies. Observations of extremophile eukaryotes in hypersaline and subglacial environments shed light on ancient survival strategies. Modern analogues, such as single-celled algae in Antarctic lakes or protozoans in brine inclusions, reveal that metabolic flexibility, protective proteins, and compartmentalization are key to surviving extreme cold and salinity. These comparisons help reconstruct how Cryogenian eukaryotes may have functioned, providing tangible evidence of strategies that allowed cellular complexity to persist under global ice coverage. Genetic and biochemical resilience allowed these cells to maintain function over centuries, despite isolation. DNA repair mechanisms countered the slow accumulation of mutations, while proteins adapted to maintain structural stability in near-freezing conditions. These strategies ensured continuity not only for individual cells, but for the collective genetic heritage of the population, preserving the foundation for evolutionary diversification once more favorable environments emerged. In their frozen microhabitats, eukaryotes quietly experimented with genetic innovation. Despite extremely slow metabolism and isolation, these cells retained mechanisms for mutation, recombination, and horizontal gene transfer, enabling gradual adaptation to their harsh environment. Such genetic activity, though limited, preserved a reservoir of evolutionary potential, setting the stage for for future diversification. These micro-scale experiments within frozen veins hint at the origins of cellular traits that would eventually allow for multicellularity and more complex ecosystems once the ice began to retreat. Within each nucleus, the machinery of life quietly endured, storing both resilience and potential for future evolution. These eukaryotic survivors carried the seeds of cooperative metabolism, organelle innovation, and genetic complexity that would ultimately enable multicellular organisms to appear. Even under the icy oppression of Snowball Earth, they demonstrate that life's persistence was not passive, it was organized, adaptive, and forward-looking. The hidden chambers of these cells hint at the cooperative networks and evolutionary ingenuity that would soon awaken in a thawing world, signaling the gradual ascent of life beyond its simplest forms. The merged and the immortal, alliances in the cold. Glacial corridors cradled the first tentative unions of cellular life. In the narrow veins of ice and brine, isolated eukaryotic cells extended toward one another, drawn by the necessities of survival. These early unions were fragile and fleeting, yet even a momentary fusion could allow organelles and metabolic components to be shared. Fossilized molecular traces in Cryogenian sediments suggest these interactions were repeated and widespread, leaving subtle imprints that reveal the first stirrings of cooperative strategies. In the harsh, frozen environment, the act of coming together was itself a statement of life's resilience and ingenuity. Cold-forged partnerships allowed individual cells to survive conditions no single organism could endure alone. When temperatures dropped to extreme lows and nutrients became scarce, cells pooled energy reserves through temporary cytoplasmic connections. Sharing mitochondria-like structures and other proto-organelles enhanced metabolic stability, allowing these partnerships to maintain minimal ATP production for survival. Modern analogues, such as extremophilic protists, provide glimpses into how cooperation could sustain life where solitary cells might perish. The advantage of collaboration in these ice-locked corridors was subtle but profound, amplifying endurance in ways individual cells could never achieve. Nuclei intertwined in ephemeral fusions, creating temporary fortresses against environmental stress. Genetic material is combined in limited recombination events, stabilizing genomes that might otherwise accumulate damaging mutations during prolonged dormancy. These interactions allowed for occasional exchanges of DNA, reinforcing key survival genes and enabling early experimentation with genome organization. The fused nuclei acted as both protective shelters and laboratories, quietly fostering genetic resilience that would later underpin more complex forms of life. Even in frozen isolation, life found a way to innovate through cooperation. Minute streams of concentrated solutes became conduits for shared metabolism and chemical signaling. Within the brine channels threading through ice, metabolites and signaling molecules could flow between connected cells, providing chemical communication and energy distribution. This network allowed clusters of cells to regulate metabolic load collectively, sustaining activity even when resources were minimal. Observations of modern halophilic microorganisms suggest these solute-mediated interactions were highly effective survival strategies. By creating micro-scale metabolic circuits, cells transformed otherwise lifeless veins of ice into dynamic, functional communities. This early cooperation magnified resilience, transforming isolated survival into collective endurance. Groups of fused or interconnected cells could withstand fluctuations in salinity, temperature, and nutrient availability that would devastate solitary organisms. Temporary organelle sharing reduced the risk of energy collapse, while cooperative exchanges maintained enzymatic activity under extreme stress. The repeated cycles of fusion and separation strengthen not only individual survival, but the emergent capacity for persistence at the group level. In these frozen corridors, survival became a shared enterprise, a precursor to more complex collaborative systems yet to come. Frozen microchambers hosted intricate patterns of connection, separation, and mutual support. Cells moved, fused, and separated in response to subtle environmental cues, forming temporary assemblies that optimized nutrient uptake and chemical balance. Cytoskeletal structures and transient bridges guided the distribution of organelles and metabolites, establishing rudimentary networks that persisted as long as conditions allowed. These microchambers were both laboratories and refuges, shaping evolutionary pathways by favoring cellular strategies that could engage in cooperation without losing autonomy. Life was quietly experimenting, using ice and brine as a framework for innovation. Periodic environmental extremes stimulated cycles of cellular merging, unlocking hidden capacities for adaptation. When solute concentration shifted or brine veins briefly warmed, cells responded by forming new fusions and reorganizing internal compartments. These cycles of interaction allowed them to buffer stress, repair damage, and experiment with new genetic or metabolic arrangements. Over time, such episodic cooperation fostered physiological plasticity and reinforced adaptive traits, seeding the mechanisms that would eventually allow multicellularity to emerge. Survival depended not on isolation, but on the ability to unite when the environment demanded it. Chemical gradients carved subtle highways through the ice, guiding the assembly and coordination of cellular partnerships. These gradients, formed by variations in solute concentration and pH, acted as invisible cues that directed cells toward each other, promoting efficient merging in areas where survival demands were greatest. Cells could detect and respond to tiny differences in their local microenvironment, optimizing fusion events to maximize resource sharing and metabolic efficiency. Some networks even developed quasi-hierarchical structures where more metabolically active cells served as hubs, distributing energy and metabolites to less active partners. This sophisticated, gradient-driven coordination demonstrates that even in the harshest frozen corridors, life found ways to organize itself, turning the ice into a dynamic landscape of biological experimentation. Cooperative networks guided organelles, amplifying cellular efficiency in niches starved of energy. By pooling mitochondria-like and other organelle analogues, cells maximized energy conversion and minimized local failures. Shared metabolic functions allowed the collective to maintain low-level activity and repair mechanisms that would otherwise have been impossible in solitary cells. These distributed networks increased resilience, effectively turning a population of fragile cells into a quasi-organism capable of persisting across decades or centuries of extreme cryogenic conditions. Cooperation became a mechanism for endurance and experimentation simultaneously. Trace biomarkers preserved in cryogenian sediments reveal the echoes of these primitive alliances. Lipids, isotopic signatures, and preserved protein sequences indicate that cellular merging and cooperative networks were recurrent phenomena rather than isolated incidents. These molecular fossils reveal that even in a world locked in ice, life actively explored strategies of connection, communication, and energy sharing. The sediments themselves serve as a record, a frozen testimony to the first experiments in cooperation that laid the groundwork for more complex life forms. Life's first experiments in cooperation planted the seeds of complex ecosystems yet to come. In these microscopic unions, evolution tested the advantages of collective survival, shared metabolism, and partial organelle exchange. the lessons learned in frozen brine veins would echo through time, forming the basis for multicellular organization and the development of higher life. Even in the depths of global glaciation, life's ingenuity persisted, demonstrating that connection could be as vital as endurance. These first acts of alliance quietly shaped the trajectory of evolution, hinting at the microbial civilizations and collaborative networks that would later dominate thawed landscapes. The architects beneath the ice. At the frozen underbelly of the glaciers, mats began to take shape, delicate yet enduring. Beneath layers of ice that stretched for kilometers, microbial communities clung to surfaces where meltwater occasionally trickled, delivering the tiniest doses of nutrients. These thin films of life were invisible to the naked eye, yet their persistence marked the beginning of structured ecosystems in the harsh Cryogenian cold. Using biochemical tools, scientists have identified remnants of these ancient mats in glacial deposits, revealing the earliest examples of life constructing its own microhabitats beneath impenetrable ice. The quiet endurance of these communities set the stage for more elaborate ecological architecture. Layer upon layer, microbial communities secreted compounds that mineralized into the earliest stone records of life. Calcium carbonate, silica, and iron-rich minerals were excreted by microbes, gradually building visible laminae that could survive for millions of years. These deposits preserved not only the shapes of cells, but also subtle chemical signatures of their metabolism, encoding ancient interactions in stone. Researchers examining Cryogenian stromatolite analogues have measured layer thicknesses and chemical composition, confirming that even in near total darkness, life could leave a permanent imprint on its environment. These mineralized layers were the first durable monuments of survival in a world otherwise dominated by ice. Even under scarce sunlight and low oxygen, chemical gradients guided growth and organization within the biofilms. Microbes detected minute differences in solute concentration, pH, and redox potential, arranging themselves along these gradients to maximize access to energy sources and nutrients. Some communities aligned along microchannels where oxygen or sulfide levels fluctuated, creating patterned mats that optimized metabolic efficiency. Such gradient-guided structuring demonstrates that even microscopic organisms could organize themselves spatially to survive extreme and variable conditions. The ice, once a barrier, became a scaffold for life to innovate. Filamentous structures intertwined, forming networks that stabilized both sediment and microbial activity. Cyanobacterial filaments and other microbial threads wove through the mats, creating frameworks that reduced erosion and retained water pockets. These networks enabled prolonged metabolic activity during periods of extreme cold, preventing communities from being entirely immobilized. Studies of modern analogues in polar and hypersaline environments show that filamentous mats maintain nutrient flow and structural integrity, suggesting that Cryogenian mats relied on similar principles to thrive. Life was building not only for survival but also for permanence. Periodic pulses of meltwater nourished these colonies, delivering essential nutrients and ions in fleeting windows. Seasonal or episodic melting created short-lived channels of liquid water, carrying dissolved minerals that fueled growth and chemical reactions. Microbes quickly absorbed these transient inputs, converting them into biomass and secreting compounds that reinforced mineral layers. Each pulse was brief, yet over centuries and millennia, these repeated events produced measurable expansion of mats and enhanced the structural complexity of the colonies. Even in a frozen world, life found ways to capitalize on ephemeral abundance. Water-sculpted microchannels enabled the circulation of vital compounds, sustaining richly layered chemical ecosystems. These tiny conduits acted as lifelines, distributing dissolved carbon, nitrogen, and sulfur compounds between distant parts of the mat. Cells positioned near the channels could access fresh resources, while others recycled waste products, creating localized cycles of metabolism that increased overall community efficiency. Analysis of modern analogues in polar environments demonstrates that such channel networks enhance resilience, allowing mats to survive prolonged periods of darkness and low oxygen. Over time, these circulating flows also promoted subtle structural reinforcement, as microbial secretions bound minerals along the channels, contributing to the early fortification of the layered colonies. Mineral deposition created subtle topographies, preserving patterns that would survive millions of years. As microbes excreted calcium, silica, and iron compounds, small ridges, domes, and layered textures emerged, forming a kind of topographical fingerprint of biological activity. Modern imaging techniques reveal microlaminae within cryogenian deposits, where alternating layers correspond to metabolic cycles and environmental fluctuations. These stone records not only document the presence of life, but also provide insights into how early ecosystems adapted to extreme cold, low light, and oxygen scarcity. Interactions between bacterial species enabled division of labor, with some fixing carbon while others recycled nitrogen or sulfur. Specialized metabolic niches allowed microbial mats to maximize resource efficiency, maintaining local nutrient cycles even under stress. Certain bacteria acted as chemolithotrophs, drawing energy from chemical compounds in the rock and ice, while others utilized photosynthetic or heterotrophic strategies where faint light or organic material was available. The interdependence of these microbial guilds illustrates that complex ecological relationships were emerging long before multicellular life appeared, highlighting the sophistication of even microscopic communities. Ancient layers of stromatolite analogues hold the encoded traces of life's metabolic rhythms. Microscopic inspection of ancient layers shows filament arrangements, cell clusters, and chemical signatures corresponding to carbon fixation, nitrogen cycling, and sulfur metabolism. Each layer is a testament to endurance, recording the rhythms of microbial life that persisted under glaciers for thousands of years. Geochemical analyses of these formations provide a window into the strategies life used to survive isolation, low oxygen, and minimal nutrient flux, preserving evidence that would guide our understanding of early ecosystems. Slow, patient growth transformed ephemeral microbial films into persistent ecological fortresses beneath the ice. Over centuries, mats thickened, stabilized, and became increasingly resilient to environmental stress. The combination of mineralization, structural weaving, and metabolic cooperation allowed these colonies to withstand freezing, desiccation, and nutrient scarcity. These fortified mats served as miniature habitats, sheltering other microbes, retaining water, and concentrating essential ions, effectively creating entire microecosystems hidden beneath the glaciers. Life's ingenuity turned the ice into both a fortress and a record of persistence. Through these constructions, life not only survived the cold, but left a traceable signature of ingenuity for the future. The layered architecture of microbial mats would later inspire the complex ecosystems that emerged as Snowball Earth thawed. In stone, the first evidence of coordinated, communal life endures, showing that even under extreme global freezing, life adapted, organized, and engineered habitats of remarkable sophistication. These ancient architects laid the foundation for the evolutionary leaps that would follow, reminding us that survival is as much about building and shaping the environment as it is about resisting its extremes. The ones who breathed metal. When oxygen vanished, the seas grew quiet, yet chemical energy surged unseen beneath the ice. The frozen oceans of the Cryogenian were dominated by darkness and silence, yet life persisted in hidden pockets where dissolved metals and gases offered alternative pathways for survival. Microbes had long relied on oxygen, but as levels plummeted beneath the ice, evolution compelled them to innovate. Modern studies of iron and sulfur-rich sediment cores revealed traces of microbial activity from this period, suggesting that life had shifted its breathing to chemical elements that would previously have been toxic or irrelevant. In these invisible chemical battlegrounds, survival depended on exploiting every potential energy source. Iron ions became conduits for respiration, allowing microbes to extract energy in the absence of oxygen. Ferrous and ferric compounds served as electron acceptors, driving metabolic reactions that sustained cellular functions. These iron-respiring microbes left distinct chemical signatures detectable in later sediment layers as isotopic anomalies. Laboratory experiments with modern analogues demonstrate that even minimal iron availability can fuel metabolism, confirming that cryogenian microbes could persist under extreme scarcity. The oceans may have been frozen solid above, but beneath, cells thrived by turning metal into breath. Sulfur-rich pockets created microhabitats where bacteria could flourish, cycling compounds in dark, frozen waters. Hydrogen sulfide and sulfate provided both energy and chemical niches, allowing microbial communities to metabolize efficiently despite minimal external nutrients. These sulfur-respiring organisms created localized chemical microenvironments that buffered extreme conditions, stabilizing pH and ion concentrations within their mats. Sediment cores from glaciated regions show layered sulfur deposits interspersed with microbial biomarkers, offering a window into the spatial organization and ecological sophistication of these communities. Life adapted not by retreating, but by mastering the chemistry of its surroundings. Methane seepages acted as alternative energy sources, feeding communities adapted to the chemical shadows. Tiny cracks in ice or rock, sometimes released, trapped methane, which anaerobic microbes could oxidize for energy. These cells often partnered with others, forming consortia where one species converted methane into intermediate compounds that another could utilize, creating localized food webs. Modern methane seeps in polar regions provide living analogues, showing how such cooperation enhances survival in extreme conditions. Even in a frozen world, energy flowed in hidden channels, sustaining delicate yet resilient ecosystems. Enzyme systems evolved to harness these metals and gases, reflecting a metabolic ingenuity unmatched in warmer times. Specialized proteins enabled the extraction of electrons from iron, sulfur, and methane, driving ATP synthesis in oxygen-free environments. Genetic studies of extant extremophiles reveal homologous enzymes, suggesting a deep evolutionary continuity from the Cryogenian. These biochemical adaptations were not isolated but spread through microbial networks, allowing life to explore a breadth of chemical possibilities previously inaccessible. Evolution, in these moments, was chemical as much as structural, rewriting the rules of existence beneath the ice. Trace isotopic signatures preserved in sediments reveal patterns of metal respiration across the Cryogenian. Variations in iron and sulfur isotopes point to localized microbial processing, indicating that communities were widespread and metabolically active despite global glaciation. These signatures also help reconstruct the timing and intensity of chemical adaptations, showing how life responded to fluctuating oxygen levels. Each layer of rock, each isotope anomaly, is a testament to survival strategies that balanced chemical opportunity with environmental risk. Communities clustered around mineral veins, forming dense networks of chemically linked cells. Iron and sulfur-rich veins became hotspots of microbial diversity, as cells congregated to exploit high concentrations of usable compounds. Within these networks, cells communicated through chemical signaling and exchanged metabolites, creating primitive cooperative systems that magnified collective resilience. Cryogenian mats and filaments preserved in ancient rocks suggest that these networks were spatially structured, with high-density hubs surrounded by sparser peripheral cells, maximizing the efficiency of resource use. Life was no longer merely individual, it had begun to function as a community bound by chemistry. Subtle gradients of electrons and ions streamed through the mineral veins, guiding microbial metabolism along invisible paths. These microscale currents allowed certain cells to specialize in extracting energy from specific elements, while neighboring microbes captured byproducts and redistributed them to maintain chemical balance. High-resolution studies of modern analogues show that such electron-conducting networks improve energy efficiency and enhance survival under extreme nutrient limitation. In the Cryogenian, this meant that even a single mineral vein could support complex, cooperative ecosystems, with cells dynamically adjusting their metabolic roles as environmental conditions shifted. These invisible circuits of energy flow not only stabilized microbial networks, but also created a template for later ecological innovation, demonstrating that chemical organization could rival physical structures in shaping life. Some microbes partner symbiotically, exchanging electrons and metabolites to stabilize energy extraction. Direct electron transfer between species allowed communities to survive where solitary cells could not, especially in nutrient-poor or fluctuating chemical conditions. These partnerships are reflected in modern syntrophic consortia, which trace their evolutionary roots to cryogenian chemical ecosystems. Metabolic specialization created dependencies, ensuring that diverse species persisted together, strengthening overall resilience. Even in total darkness, life invented collaboration as a strategy for endurance. The diversity of chemical breath laid the foundation for future metabolic flexibility and ecosystem complexity. By mastering iron, sulfur, and methane respiration, microbes expanded the possible niches they could inhabit, establishing the groundwork for later ecological innovation. Communities that survived the icehouse set evolutionary precedents, showing that adaptability could thrive even under global freezing. Life's creativity in chemical metabolism was a silent but profound force, quietly shaping the potential for multicellular and more complex forms in subsequent eons. In these hidden metabolic worlds, evolution quietly accelerated, preparing life for eventual expansion as the ice thinned. Chemical ingenuity, symbiotic networks, and spatial organization all contributed to a resilient biosphere that would survive the harshest conditions Earth had ever experienced. While the planet remained largely frozen, these microbial pioneers silently rewrote the blueprint of survival, demonstrating that even in isolation, life finds ways to innovate. Beneath the frozen seas, evolution had discovered not only survival, but opportunity. And in these chemical lungs, the future of Earth's biosphere was quietly being prepared. Evolution in the dark, under layers of unyielding ice, life did not pause, but continued its quiet experiments in adaptation. Even in the absence of light, warmth, and open seas, cells persisted, subtly reshaping themselves in response to frozen isolation. Genetic variation, though slow in such harsh conditions, became the engine of survival, enabling some lineages to withstand extreme chemical and thermal stress. Sediment cores from Cryogenian glaciated regions show patterns of isotopic anomalies that hint at active, metabolic, and evolutionary processes, indicating that life continued its quiet, relentless push forward even beneath the planet's frozen shell. Mutations arose in isolated cells, some conferring subtle advantages in nutrient capture or chemical metabolism. Minute genetic changes allowed microbes to exploit microgradients in ions, metals, and solutes, maximizing energy extraction where resources were sparse. Modern analogues of extremophiles illustrate how single nucleotide substitutions can lead to enhanced enzyme efficiency or tolerance to toxic compounds, suggesting that Cryogenian microbes may have undergone similar microevolutionary shifts. Each mutation, though individually small, contributed incrementally to the broader tapestry of survival, quietly favoring those best suited to endure the frozen abyss. Microbial lineages diverged along frozen channels, exploiting the smallest gradients in ions and solutes. Brine veins, fissures in ice, and mineral-rich pockets became laboratories of evolutionary experimentation. Isolated populations slowly differentiated, accumulating unique adaptations tailored to their local microhabitats. Fossilized microbial mats and isotopic analyses suggest these segregated communities were metabolically diverse, each lineage discovering its own chemical niche and refining its strategies for energy acquisition, resilience, and reproduction. Long periods of stasis were punctuated by bursts of innovation when chemical or thermal anomalies appeared. Occasional thawing events, geothermal intrusions, or shifts in brine composition created short-lived windows of opportunity, prompting rapid evolutionary adjustments. Microbes responded by expanding metabolic repertoires, enhancing stress responses, or developing cooperative behaviors with neighboring cells. These episodic accelerations of change allowed life to explore new pathways, leaving enduring genetic footprints that persisted long after the anomalies faded. Genetic evidence preserved in Cryogenian sediments hints at the early emergence of regulatory networks that controlled cell division and stress responses. Molecular fossils suggest that even in extreme cold, cells began coordinating internal processes with remarkable precision. These networks regulated the timing of replication, energy allocation, and protective mechanisms against freezing and chemical toxicity. Such regulatory sophistication was crucial as it enabled microbes to survive prolonged periods of scarcity and environmental unpredictability, laying the groundwork for the eventual rise of more complex eukaryotic cells. Communities in deep fissures and brine pockets evolved cooperative behaviors, increasing collective resilience. Symbiotic interactions, metabolite sharing, and coordinated chemical signaling became vital for maintaining viability under extreme constraints. Cells that partnered with neighbours could stabilize local chemical environments, reduce waste accumulation, and enhance energy efficiency, effectively creating primitive microecosystems. These collaborative networks foreshadowed the ecological interdependencies that would dominate life after the icehouse, demonstrating that evolution favoured not just individual fitness, but also collective adaptability. Hidden exchanges of DNA threaded through microbial communities, linking distant cells in a web of shared innovation. Horizontal gene transfer allowed advantageous traits to spread rapidly, even between unrelated lineages, bypassing the slow pace of mutation. Plasmids, transposons, and viral vectors carried genes for stress tolerance, metabolic versatility, and chemical sensing across isolated microhabitats. Evidence from modern analogues of extremophiles shows that such genetic sharing increases resilience and promotes adaptive radiations, suggesting that cryogenian microbes were not only evolving individually but also collaboratively. These subtle genetic currents enabled populations to respond to extreme fluctuations in solute concentrations, temperature gradients, and energy availability, reinforcing the evolutionary dynamism of life beneath the ice. Slow but persistent adaptation allowed microbes to explore novel metabolic pathways even under extreme scarcity. New enzymes, electron acceptors, and chemical reactions emerged, giving cells access to previously untapped energy sources. Iron, sulfur, and methane respiration continued to diversify, but cells also experimented with more subtle chemical interactions, such as electron shuttling and microscale nutrient recycling. These adaptations increased the range of viable niches, ensuring that life could maintain continuity across the most hostile stretches of ice-covered oceans. Comparisons with modern extremophiles reveal a continuity of survival strategies that traces back to this frozen age. Organisms living today in Antarctic subglacial lakes, deep-sea hydrothermal vents, and hypersaline brines exhibit metabolic, structural, and behavioral strategies reminiscent of cryogenian microbes. Molecular studies link certain genetic pathways, such as stress response proteins and anaerobic respiration genes, directly to lineages that likely evolved during Snowball Earth. These living fossils offer a window into the ingenuity of evolution in isolation, confirming that adaptation under extreme conditions is not only possible but enduring. Incremental evolutionary changes accumulated over millennia, quietly preparing the ground for the post-Snowball diversification of life. While the world above remained a frozen desert, cells below were building genetic repositories of resilience, experimentation, and innovation. Each minor adaptation, each cooperative network, and each new metabolic strategy contributed to a foundation upon which later, more complex ecosystems would be constructed. The seeds of multicellularity, ecological specialization, and biochemical diversity were sown in the ice, invisible yet profoundly influential. Beneath the frozen crust, evolution had written a subtle but enduring legacy, awaiting the planet's eventual thaw. Life's silent pulse persisted, threading together the fates of isolated lineages across brine veins, mineral networks, and subglacial fissures. Even in near total darkness and near absolute cold, the relentless drive of mutation, selection, and cooperation ensured that biology did not merely survive, it quietly innovated. In the depths of the Snowball Earth, evolution continued its inexorable march, and the blueprint of future life quietly unfolded, prepared for the moment when light and warmth would return. The heartbeat beneath the ice. In the frozen darkness, life's pulse continued, imperceptible but steady across isolated niches. Beneath miles of unbroken ice, microbial communities persisted in a rhythm almost too subtle to detect, yet it carried the unyielding energy of survival. Even as sunlight vanished and temperatures plummeted, cells maintained metabolic processes, cycling nutrients, and storing energy in brine-filled fissures. Chemical gradients in subglacial veins guided movement and metabolism, allowing life to thread together a network of continuity. Sediments from cryogenian deposits record these pulses indirectly, preserving isotopic signatures that testify to an ecosystem quietly enduring through geologic time. Salt-laden veins carved through ice and rock carrying nutrients to waiting communities. These liquid highways, enriched with ions and dissolved minerals, functioned as arteries for the frozen biosphere, connecting isolated microhabitats across vast distances. Microbial life exploited these channels to share metabolites, exchange genetic material, and distribute energy efficiently. Geochemical studies suggest that concentrations of sodium, magnesium, and calcium fluctuated slowly, offering predictable cues that microbes adapted to over millennia. Even minor shifts in these veins could ripple through the frozen networks, prompting subtle changes in growth and activity. Biofilms formed layered networks, linking generations through chemical signaling and shared metabolism. Communities adhered to rock surfaces or the undersides of ice, creating stratified mats in which different microbial species occupied distinct niches. Enzyme gradients, exopolysaccharide layers, and redox-sensitive compounds orchestrated interactions between layers, allowing cells to cooperate despite extreme scarcity. Fossilized microbial mats in Cryogenian sedimentary structures reveal mineralized traces of these early fortresses, hinting at organizational sophistication previously unseen in such extreme conditions. Each mat was both shelter and laboratory, preserving life's continuity while testing metabolic innovations. Microbial activity oscillated in response to subtle thermal and chemical variations within the ice. Geothermal heat, localized brine enrichment, or transient solute pulses created periodic windows of higher metabolic activity. Cells responded with synchronized division cycles, energy allocation, and protective metabolite production. These oscillations were not uniform, but they created a quasi-rhythmic pattern that reinforced survival across generations. Isotopic measurements from embedded sediments show these cycles left discernible chemical traces, offering modern scientists glimpses into life's steady cadence beneath the ice. Even in isolation, populations exhibited collective regulation, maintaining equilibrium across microscopic ecosystems. Density-dependent feedback mechanisms, quorum sensing, and cooperative metabolite recycling allowed cells to avoid overexploitation of limited resources. Single organisms acted in concert, stabilizing local environments to prevent collapse, much like larger-scale ecosystems. Modern analogues from Antarctic subglacial lakes and hypersaline brines confirm that such self-regulation is a cornerstone of microbial endurance under extreme isolation. This intricate balance illustrates how life sustained continuity, not by speed or strength, but by patience, coordination, and chemical foresight. Cells stockpiled chemical treasures, hoarding energy and protective compounds against the frozen future. Cryoprotectants, compatible solutes, and high-energy molecules accumulated in intracellular spaces, preventing ice crystal formation and sustaining enzymatic activity even at sub-zero temperatures. These biochemical reserves allowed microbial populations to endure decades, even centuries, of extreme stasis without catastrophic damage. Lipid composition shifted to maintain membrane fluidity, while specialized proteins stabilized nucleic acids and ribosomes, preserving the capacity for replication once conditions improved. Studies of modern extremophiles in polar environments show that such strategies were not incidental but fundamental adaptations, enabling the Cryogenian biosphere to persist in near-total darkness beneath miles of ice. By storing energy and protection in anticipation of eventual thaw or chemical flux, these cells transformed dormancy into a deliberate and dynamic survival strategy. Sediment cores reveal continuous metabolic activity, preserving isotopic signatures that testify to survival over millennia. Carbon and sulfur isotopes indicate ongoing respiration, fermentation, and sulfur cycling throughout the frozen ages. These records suggest that even the harshest Cryogenian winters could not halt microbial life entirely. Instead, communities slowed, restructured, and persisted in a state of minimal but sufficient activity. Molecular fossils of ancient stress response proteins further demonstrate that these microbes were equipped to endure prolonged stasis while remaining ready to exploit ephemeral resource pulses. Interactions among diverse microbial lineages stabilized energy flows, preventing local collapses despite harsh conditions. Cross-feeding, metabolite exchange, and chemical signaling created a network of dependencies in which the survival of one lineage supported the stability of others. Such relationships allowed for the persistence of metabolic diversity, ensuring that critical biochemical pathways remained functional across generations. This cooperative architecture meant that even if isolated cells succumbed, the broader network endured, perpetuating life in a frozen, interdependent equilibrium. Genetic analyses suggest that stress response pathways were conserved, ensuring survival during extreme freeze-thaw cycles. Heat shock proteins, antifreeze proteins, and oxidative stress regulators were critical for maintaining cellular integrity under fluctuating conditions. Horizontal gene transfer facilitated the spread of these protective mechanisms, linking distant populations into a functional genetic web. Fossilized biomarker evidence in Cryogenian rocks supports the presence of these highly conserved proteins, demonstrating that even under millennia of ice, evolution preserved and transmitted the tools necessary for survival. The interplay of chemical gradients and microbial metabolism created an invisible rhythm, a heartbeat that synchronized life across frozen expanses. Each brine channel, each biofilm mat, and each fissure contributed a pulse to this global biosphere. Metabolic byproducts of one community fed another, while slow cycles of energy storage and utilization regulated the pace of survival. This collective heartbeat was faint, yet its consistency over millions of years ensured that life persisted, silently preparing for a world that would one day thaw and flourish. This silent pulse foreshadowed the moment when the world above would awaken, carrying the legacy of endurance forward. Life's resilience beneath the ice demonstrates that survival does not always require abundance or warmth, but persistence, adaptability, and subtle cooperation. The biosphere's heartbeat, quiet but unbroken, embodies the tenacity of life even under conditions that seem incompatible with vitality. As Snowball Earth slowly relinquished its icy grip, these enduring microbial networks would form the foundation for a flourishing, sunlit world poised to inherit the lessons of survival written in the ice. Even under a sky locked in ice, life whispered its persistence, unseen yet unwavering, threading connections through the darkest, coldest corners of the planet. These tiny architects of survival remind us that endurance is often invisible, measured not in grandeur, but in quiet resilience and subtle innovation. As the frozen world gradually loosened its grip, the seeds of complexity, cooperation, and renewal had already been sown, awaiting the warmth to awaken them. In the lingering silence of Snowball Earth, we glimpse life's steadfast heartbeat, a pulse that would echo through every age to come. Subscribe to journey further into Earth's forgotten worlds and the deep rhythms of life.